testis. In the testis, within the testis, there are multiple small tubules. We call seminiferous tubules. Within the seminiferous tubules, the spermatogenesis occurs. What is spermatogenesis? I have already told you, there are two phases. The first phase is spermatocytogenesis or spermatocytosis, and next is the maturation, which is known as the spermiogenesis, where there is no division, only maturation. I am quick here, it is not very difficult. The primordial germ cell of the male fetus develops in the fetal period, and ultimately they form your primordial PGC or primordial germ cell. In case of a female, form oogonia. Similarly here it forms spermatogonia. What is the difference? The oogonia starts its meiotic division even before birth, but the meiosis stops at the phase of prophase 1, and when the ovum after menarche, after puberty, some ovum then go into the maturation to complete this meiosis. But the difference in the male is that they remain as a spermatogonia, no meiosis starts, because there is a meiosis inhibiting factor secreted by the Sertoli cells within the testis. You will learn later on what is Sertoli cells, which inhibits the entry of the spermatogonia into the meiosis. They stops. They remain as a spermatogonia as long as in puberty. After puberty, when situation demands the hormonal stimulation, they go into the mitosis as well as a meiosis. Mitosis as well as a meiosis. What is the mitosis? First, spermatogonia, when situation demands, when there is a male gets stimulated, then this spermatogonia, first form, I have already told you last class, convert into, after repeated division, dark type A type of spermatogonia. Dark because their cytoplasm looks dark. So dark type A spermatogonia. Dark type A. Again mitosis occurred from the spermatogonia. Mitosis. They form pale type A, pale. They are after mitosis, they give innumerable pale type A spermatogonia, which then form type B spermatogonia. So type A spermatogonia. Still gonia stage is going on. Type B spermatogonia. These spermatogonia now ultimately give primary spermatocyte. It is the largest cell of all the spermatogonia, spermatocyte present within the testes. Now what happens? Type B becomes type spermatocyte primary. Primary spermatocyte, its chromosome number, all the cases is 46, 46, 46, 46, 46. These primary spermatocyte then enter into the meiosis. Primary spermatocyte then enter into meiosis. I have already told you, meiosis has got two parts, meiosis 1 and meiosis 2. Meiosis 1 is a haploid division where the chromosome number gets up. So after meiosis 1, it becomes secondary spermatocyte. Chromosome number is 23, either 22 plus X or 22 plus Y. Some spermatocyte are 22 plus X, secondary spermatocyte, few are 22 plus Y. Then it completes quickly the meiosis 2 and becomes spermatid. Chromosome number 23 and ends the spermatocytogenesis. It takes about 40 days to complete, and here ends the spermatocytogenesis. Next, each spermatid now mature, transforms, not any mitotic division, transforms into, total shape is changed into spermatozoa. Like a tadpole become toad, right? Spermatid is a round shaped cell. It is totally round shaped with a nucleus, with a Golgi apparatus, containing granules, mitochondria, number of mitochondria, and centriole. Nucleus contain chromosome. So spermatid and other organelles are also there. So spermatid looks like that, a round cell with central nucleus containing chromosomes, Golgi apparatus, Golgi bodies, mitochondria, innumerable mitochondria, and centriole. Now when it enters into maturation or spermiogenesis, which also occurs in the testes, this shape is changed. This round nucleus goes to one pole of the cell. Goes to one end, one pole of the cell. The Golgi apparatus, which contains granules, these granules or with membranes, they form a cap. Here, they form a cap with granules. Cap covering the nucleus. Cap to be. We call acrosomal cap. Simply. And this granule is very important. This Golgi apparatus contains granules. It contains granules, which contains enzymes. High hyaluronidase, acrosome, acid phosphatase, these granules contain. So it is, this cap is nothing but formed from the Golgi apparatus, Golgi bodies. Next, mitochondria is there. innumerable mitochondria is there, and centriole. These centriole now divides into a two centriole. Centromere. One centromere now divided into two centrioles. এক থেকে দুটো হলো। And now the shape was round. The cell becomes pear-shaped. It looks like that. And one centriole goes here. And one centriole, like a ring, we also call the ring centriole, goes here. So these centriole we call the proximal centriole. This centriole we call the distal centriole. These proximal centriole then produce filaments, microtubule forming filaments, 2 plus 9. এগুলি আমি আর ডিটেলসে যাচ্ছি না। These produce filament. Centriole, already you know, from mitotic spindle in cell division, জানো তো? Mitotic spindle nothing but a tubule MP. It also produces tubular filament. Come on. And this mitochondria now comes to reside here. এই জায়গাটায় চলে আসে সব মাইটোকন্ড্রিয়া। একটু বড় করে দিচ্ছি। And cover the axial filament in spiral manner. এই জায়গাটা পেঁচিয়ে থাকে সমস্ত মাইটোকন্ড্রিয়া now comes to go down. And it becomes slightly tapering now. Right? And most of the cytoplasm now extrudes out. Only few cytoplasm are present. So, what's up? I'm now level jodi kori. It is the nucleus. It is the acrosome, cap, Golgi. The blue color is the multiple mitochondria. This is the axial filament. It is the proximal centriole. It is the distal centriole or ring centriole. This part we call head. This part, the mitochondria are present spiraling around the axial filament, we call, there is a part, small part remain, that is a neck. And this part, middle piece. And this part called tail. Tail has got a tool part. One is covered by a fibrous membrane. Called principal piece. And this is the end piece. So this is the mature spermatozoa, which is contain a head, a narrow part neck, then middle piece and tail. Tail consists of a principal piece above proximally and distal piece known as the end piece. All are covered by a cytoplasmic membrane. Obviously, a cell is covered by a cytoplasmic membrane. This is the mature sperm. And this process takes about 24 days to complete, and this process is called spermiogenesis within the testes, and still they are not motile, they are immotile, and when ejaculation occurs, the millions of sperm now goes to from the testes goes to a tubule known as the epididymis. While in the epididymis, they get some glycoprotein coat, towards the acrosomal cap, and also some seminal proteins, and attain maturity. So in the testes, the spermatozoa are not motile. When they ejaculated, when they come from the testes, where they go? From the testes, এখানে produce হচ্ছে, they go, and this is the epididymis. While during the passing to the epididymis, they get a covering of the glycocalyx, glycogen and protein covering, coating, and also other seminal vesicles, and ultimately then they pass from the epididymis, they goes to the vas deferens, then to the ejaculatory duct, then to the urethra, they comes out. And during this process, the seminal vesicle through the ejaculatory duct, another organ, as an accessory sex organs present within the male, they also acts prostaglandin, etc., and other important enzymes. Those sperm are now fully matured and they are ejaculated. So, when the spermatozoa gets the fluid of the secretion of prostate, seminal vesicles, etc., we call semen. So, millions of spermatozoa during ejaculation, while passing through the male genital tract, passing through the vas deferens, passing through the urethra, they attain maturity, and ultimate outcome is the semen. Right? And during it is being found per ejaculation, 39 million sperms are ejaculated. 39 million. Around 40 million. 39 million sperm per ejaculation, not per ml, per ejaculation. About 2 to 3 cc, range is 1.5 to 2.5 cc. So seminary ejaculate 39 per ejaculator ejaculated. So it is the final process. So I have given you. So finally, sperms are ejaculated, and if during sexual intercourse, these sperms are ejaculated into the vagina of the female. Similarly, the Graafian follicle also matures within the ovary. At about 14th day of menstruation period, this mature Graafian follicle burst out from the ovary and taken up by the fallopian tube. Right? Fallopian tube. And by their slight movement and the tubal contraction of the muscles of the fallopian tube, these mature ova now reside and wait for sperm at about 24 to 72 hours, or in some books written 24 to 48 hours within the fallopian tube. And if spermatozoa reach within this period, there is a chance of fertilization, right? And when the Graafian follicle ruptures and the mature ovary goes, what does it take? What does it take? It is the mature oocyte with a nucleus containing 22 plus X chromosome, 22 plus X chromosome. The plasma membrane of the oocyte is called vitelline membrane. We call vitelline membrane. Plasma membrane of the oocyte is called vitelline membrane. Surrounding it, there is a zone of glycoprotein coat. I have already told you, this is the zona pellucida. Zona pellucida. There is a gap between the zona pellucida and vitelline membrane. What is this space called? Perivitelline space. What in the perivitelline space? What is present? First polar body. When the mature ova starts a burst, it completes its first meiotic division. That's mean reduction division. In the reduction division, the two cells are produced. One cell is large, containing all the nutritive material within its cytoplasm, which is the mature oocyte, mature ovum. And another is small, very negligible, we call polar body, which remains in the perivitelline space. And outside and this is the very important barrier known as the zona pellucida. Zona pellucida containing some receptor, ZP2, Z. And surrounding the zona pellucida, there are 2 to 3 layers of cumulus oophorica cell we call corona radiata. So it is the mesothelium, it is vitelline membrane, it is zona pellucida, and it is the corona radiata. They present within the ample of the fallopian tube, waiting for the sperm to fertilization, for fertilization. And the sperm is ejaculated. About 39 million sperms are ejaculated within the vagina. They must travel to the uterus, fallopian tube. And they travel. How they travel? Number one, I have already told you, seminal vesicle contributes prostaglandin to the semen. বলেছি এই মাত্র seminal vesicle also contributes some chemicals known as the prostaglandins, which are adds to the semen. So sperm is remain within the semen. Semen is ejaculated. This prostaglandin, when comes into the contact of the female genital tract, especially uterus, they stimulate the uterine musculature. The uterine muscle has got three coats: endometrium, muscle bolche na, uterine wall has got three coats: endometrium, myometrium, and perimetrium. Myometrium means muscles. This muscle starts contraction. And also, during intercourse, in the female, oxytocin is secreted by the posterior pituitary. They also stimulates the muscle contraction. So there occur uterine muscle contraction. So uterine muscle contracts in this way, produce a vacuum within the upper part of the cavity of the uterine cavity, so that some sperms are sucked out in. In the uterus, the muscles are contracting, so there is a vacuum there. This is a positive pressure. So the semen are sucked in, the spermatozoa are sucked in the upper part of the uterine cavity. Don't tell, due to the tail movement of the sperm, they swim. No. The movement by the uterine musculature, action. And they ultimately goes to the so they goes to the either one or either one of the fallopian tube. And during this, in the fallopian tube, the contraction of the fallopian tube and the ciliary movement, the sperms gradually goes there in the ampulla. How long it takes? 2 to 7 hours. 2 to 7 hours. Right? So if the time, if it occurs, then and then fertilization occurs. Then sperm, but it has been found during this movement, 39 million sperms are ejaculated. Only 300 to 400 sperms are able to go to the ampulla of the fallopian tube. Only 300 to 400. Out of 300 to 400, only one spermatozoa is able to fertilize the egg. One spermatozoa, one egg, normal. If more than one, that is an abnormal. Right? And during this process, while the spermatozoa traveling this fallopian tube from here to here, there occur further maturation of the spermatozoa. Changes occur. We call capacitation. We call capacitation. Kokona in the uterine tube. Capacitation of the sperm. Capacitation. Capacitation of the sperm. Where this glyco, I have already told you, this is a glycoprotein coat and this coat has removed. Because if this glycoprotein coat is present, sperm will not be able to contact with the vital end membrane, cannot fertilize. So during this capacitation, this glycoprotein coat is removed. So sperm is now come, here is the ovum, here is the ovum and multiple sperm. Multiple sperm. Multiple sperm. 300 to 400 sperm. multiple sperm able. Now all the sperms are one to fertilize, but only one is able, যে দৌড়ে ফার্স্ট হবে। এবং তাহলে এই যে fertilization occur, now the chapter is now fertilization. What is fertilization? Is a process by which male and female gametes are fused together. So it is a anti-process of cell division. In cell division, one cell becomes two. But in the fertilization, two cells become one. So it is a fusion, not fission. So it is a fusion. Cell division e ki hoy? Ekta cell theke duto, duto theke charte. But here two cell, one ovum and one spermatozoa become fused together to form a single cell called zygote. Now, আমরা কি বলি? What is the now the fusion of the sperm? তাহলে আমরা fertilization তিনটে হেডসে ভাগ করি। Movement of the sperm, movement of the ovum into the ampulla. Number 2, fusion of the gamete. Number 3, most important, effect of fertilization. Fertilization, it is also included within the fertilization. Fertilization doesn't mean only fusion হয়ে গেল আমি লেখা শেষ করে দিলাম। No, if fertilization short note comes, you have to write effect of fertilization. After fertilization, what happens? তাহলে movement I have already told you. Next, fusion. Now question is that fusion of the sperm with the spermatozoa, what are the barriers? What are the barrier? কি বাধা দেবে? Number 1, corona radiata. এই ঢুকতে হবে এটাকে ভেদ করে। এটা একটু pierce the corona radiata so that it can enter. Number 2, barrier. Barrier মানে বাধা, obstacle, basement membrane. Sorry, zona pellucida. Sorry. Next barrier is the zona pellucida. A third barrier is the vitelline membrane. So these are the barriers of fertilization, fusion of the gametes. What's happened? কি করে তাহলে barrier হবে? All of the sperm touches this. One sperm, now when this sperm it touches this corona radiata, acrosome containing granules which contains high allurinidase and acrosine like matter which includes acid phosphatase, right? These acrosine granules now liberated and makes a whole and dissolutes মানে কি গলিয়ে দেয়, ধ্বংস করে দেয় the corona radiata. এবার ভাবো, there are so why these 300 and 400 sperms are needed comes here. Only if the high allurinidase produced by the one sperm, it is very minimum, microCC. It can penetrate only a few area of the corona radiata. So what's happened? All the sperm touches and all the sperm contributes to make a pierce in the corona. It destroys the corona radiata. কে destroy করছে? Acrosome granules. Acrosome granules, where does it present? Within the acrosome. What is acrosome? Acrosome is made up from the Golgi apparatus of the spermatozoa. And where these cortical granules are released from the spermatozoa, we called it acrosome reaction. We call acrosome reaction. So while the sperms are touching the corona radiata, the corona radiata touch করলো, this sperm now touch the zona pellucida. So while it touching the zona pellucida, it releases the acrosome granules which not only dissolutes the corona radiata, destroy করে দেয়, make a hole, also make a holes in the part of the zona pellucida. Not all zona pellucida will disappear. Zona pellucida will remain. ভালো করে শোনো, after 5 days after fertilization, না থাকলে বিপদ হতো। Only one of them, one of the sperm. Many sperms are required to solute the corona radiata cells, right? But one sperm able to dissolve the zona pellucida, make a hole and touch the vital membrane and anchor to them. And he is very selfish. He is very selfish. Only one sperm, সে সবচেয়ে বেশি দৌড়াতে পারে, সে সবচেয়ে বেশি এবল, সবচেয়ে বেশি motile, সে touch the vital membrane. যেই touch the vital membrane করে, সে স্টিমুলেট করে, ওভামেও এখানে গ্র্যানুলস আছে। এখানে যেমন acrosomal গ্র্যানুল বলি, এই গ্র্যানুলসকে বলে কর্টিক্যাল গ্র্যানুলস। তাকে কি বলি? কর্টিক্যাল গ্র্যানুলস বলে। So only one sperm is able to touch the vital membrane. যে touch করে cortical granules are released. Cortical granules are released within this perivital space, and it makes the vital membrane impermeable to other sperm. যেই cortical granules are একটাই টাচ করলো জোনা পেলুসিডাকে, এই ভাইটাল মেমব্রেনকে, করেই ও কাজ করলো, ওভামকে বললো তাড়াতাড়ি গেট বন্ধ করো, কুইকলি ক্লোজ দ্য ডোর। সাথে সাথে ও সব গেট তো বন্ধ করলো, চারপাশে বন্ধ করে দিল যাতে অন্য কোন স্পার্ম আর ঢুকতে না পারে। ইট প্রিভেন্টস পলিস্পার্মি, যাতে মাল্টিপল স্পার্ম আর না ঢুকতে পারে। ক্লিয়ার? এই কারণে একটাই স্পার্ম করলে তাহলে এই টোটাল প্রসেসটা হচ্ছে ফিউশনে লাগে। এরপরে দেখা গেছে এখানে একটু ভ্যারিয়ে আছে, এরপরে কিন্তু দেখা গেছে স্পার্মের শুধু হেড পার্ট and part of the tail can enter within the ovum, not the middle piece and other. ভালো করে বোঝো, স্পার্মের কি কি পার্ট রয়েছে? স্পার্মাটোজোয়ার হেড, নেক, মিডল পিস and tail. Only head and part of the tail can enter. অনেক বইতে টেলটাও বলে না। ধরে নেই only head can enter within the cytoplasm of the ovum containing male 22 plus X or 22 plus Y chromosome, right? তার জন্য তোমাদের শরীরে যে মাইটোকন্ড্রিয়া আছে, all are contributed by the mother, not by the father. কারণ স্পার্মের মাইটোকন্ড্রিয়া ঢুকতে পারেনি, কারণ মিডল পিস ঢোকেনি। তাহলে কি হলো? এইবারে স্পার্মের হেডটা ঢুকে গেল। যে টাচ করলো এই একটা স্পার্ম, কুইকলি ovum completes its second meiotic division. So after fertilization, at the time of fertilization, when the spermatozoa touches the vital membrane, ovum completes its second meiotic division and it becomes another polar body is here and the one remain here. So when it completes the ovum completes its second meiotic division, MCQ, at the time of fertilization. When completes its first meiotic division, at the time just before ovulation. মনে থাকবে, just before ovulation, ovum contributes its first meiotic division, and at the time of fertilization, ovum contributes the second meiotic division. Right? Now, the male nucleus, head and female nucleus are present. Then the nucleus of the male becomes swollen, or head of the sperm becomes swollen. We call it male pronucleus. And female nucleus is also swollen. We call female pronucleus. ফুলে যায়, বড় হয়ে যায়. And then the fusion of this chromosome of the male and the female occur, resulting into 46 chromosome. And it is the completion of the second part of fertilization, fusion of the gametes. third part of fertilization, after effects of fertilization. Number one, restoration of the diploid number. That's mean restoration of the 46 chromosome. এতক্ষণ মেলের ছিল 22 plus X or Y, ফিমেলের ছিল 22 plus X. তাহলে কি হলো? Restoration of the 46 chromosome. Number two, now we call the final fused cell zygote containing 46 chromosome. Number three, it determines the sex of the progeny. If it is a 22 plus X of the sperm fused, it becomes female. If 22 plus Y fused or fertilizes the female, it becomes male. Number three, last but not the least, it initiates the cell division of the zygote immediately. We call cleavage division. We call cleavage division. We call cleavage division. Cleavage division. Cleavage. এরপর একটু ছবি দেখি আর আপনাদের দেরি হয়ে যাবে, তাড়াতাড়ি যেতে হবে, খুব একটা বড় ব্যাপার নয়। এই যে restoration of the diploid number of chromosome, determination of the sex of the embryo and initiation of the cleavage division. যেটা একটু বলে দিই, তোমাদের প্রফেসর পড়ানোর কথা পড়িয়েছি কি? অ্যাসিস্ট তোমাদের কিন্তু কম্পিটেন্সিতে আছে, আমাদের এতদিন ছিল না, এবারে ঢুকিয়েছে। আছে কি? পড়িয়েছেন কি অ্যাসিস্ট্যান্ট ইয়ারাই? তাহলে একটু বলে নিই, পরের দিন পড়বো। About 1 in 6, খুব শর্টে, যাতে একটা ছোট্ট শর্ট নোট দিলে লিখতে পারো, কারণ এখন টুডে আমরা দেখছি ওরা কম্পিটেন্সি দেখছি আর কোশ্চেন দিয়ে দিচ্ছে, কেমন? এবারে কিন্তু দিয়েছে, আমি কেন এত ডিটেলস পড়ালাম, এইবারে দিয়েছে, কেমন? About 1 in 6 couples have difficulty in conceiving, having birth. About 90% of the infertile couples can conceive with medical intervention. আগেকার যুগ আর নেই, আমরা যদি অ্যাসিস্ট করি, medical doctors can assist, about 90% of the infertile couple can produce baby. Methods of artificial reproduction technology. Counseling, অনেক সময় জানে না যে when to make an intercourse. 14 day তে ওভুলেশন হয়, তার প্লাস মাইনাস 2 to 3 days যদি ইন্টারকোর্স করা যায় তবে ম্যাক্সিমাম চান্স অফ ফার্টিলাইজেশন হবে। At the end, just after menstruation, just before menstruation, usually may happen, rare or not. তাছাড়া আরো আছে ওগুলি কাউন্সিলিং। নেক্সট সাম মেডিকেশন, যাদের ওভুলেশন কম হয় তাদের there are some medication, protein, citrate, etc., also gonadotropin hormones normally can produce fertile. আমরা যেটা পড়বো IVF, in vitro fertilization। এছাড়াও রয়েছে গ্যামেট নাউডেজ মোর অ্যাডভান্স হয়েছে, ইন্ট্রাসাইটোপ্লাজমিক আছে কি লেখা নেই, fallopian tube transfer, gamete intrafallopian transfer, zygote intrafallopian transfer। মানে কি, জাইগোট আমরা বাইরে জাইগোট তৈরি করে নিচ্ছি, এই জাইগোটটাকে ফ্যালোপিয়ান টিউবে দিয়ে দিচ্ছি, এবারও নর্মালি হবে, কেমন? যেটা ইম্পর্টেন্ট এটা একটু জেনে নাও। The first baby of the in vitro fertilization was Louise Brown, 1978, 25th July। ব্যাড লাক আমাদের একজন আছে, তোমরা জানো, একটা ভালো সিনেমা has been done, এক ডক্টর কা মৌত, with the Subhash Mukhopadhyay of the National NRS Medical College, also pioneer to have a success to produce the test tube baby, but bad luck, he did not রেকর্ড, একটা যে রিসার্চ করলেই হলো না, তোমাকে রেকর্ড রাখতে হবে, প্রুভ করতে হবে, ইউ হ্যাভ টু প্রুভ ইয়োরসেলফ দ্যাট আই হ্যাভ ডান প্রপারলি। রেকর্ড রাখেননি, যখন চ্যালেঞ্জ বাই দি ওয়ার্ড ইট ডিড নট প্রডিউস and ultimately he committed suicide. If time permits or available, you must visit a cinema, possibly by Tapan Sinha, Ek Doctor Ka Maut, a short film, Ek Doctor Ka Maut, right? বইটা দেখো তোমরা, প্রচন্ড ভালো বই, হ্যাঁ, এবং সেখানে সুভাষ মুখোপাধ্যায়কে ডিপিক্ট করা হয়েছে। তাহলে কিন্তু আমাদেরই নোবেল প্রাইজটা হতো। সুভাষ মুখোপাধ্যায় ব্যাড লাক করেননি, he's been tortured by the that time government also. আচ্ছা, ভেরি ইজি, অত ডিটেলস এর মধ্যে লেখার দরকার নেই, ব্যাপারটা কিছু নয়। sperm can be ovum. How do you collect the ovum? First, initially you give some gonadotrophic stimulating, ovum stimulating factor like clomiphene citrate, gonadotropin releasing hormone, etc., so that multiple ovum can produce and mature properly. And counting the time at the about 14th day of ovulation, 14th day of menstruation, we put a nick and put a endoscope known as a laparoscopy within the abdominal cavity, and easily we can collect the mature ovum within the laparoscopic tube, right? তাহলে ওভাম আমরা কি করলাম, কালেক্ট করলাম, এখান দিয়ে একটা ফুটো করে এন্ডোস্কোপ টিউব দিয়ে সাকশন করে কালেক্ট করে নিলাম, দেখা যায়, এবং ওখানে ক্যামেরা থাকে, ভিজুয়ালাইজ হয়, প্রপারলি দেখা যায়। and this ovum is now put in a petri dish and cultured in a proper environment and kept there. And sperm, already you know, sperm from his husband or male partner or donor sperm, now put into the dish, petri dish, on the close vicinity of the ovum. As it is occurs outside the body of the female, we call in vitro. In vivo means within the body. আমাদের বডিতে যে ঘটনা ঘটে তাকে আমরা বলি ইন ভিভো, আর আউটসাইড যদি হয় তাকে বলা হয় ইন ভিট্রো। So as this fertilization occurs outside the body, we call in vitro fertilization. But proper environment, freezing and other apparatus, proper chemicals are very necessary, otherwise it will be destroyed. Then the fertilization occurs, and they want to do more than 2 to 3 ova are collected, and fertilization they occur more than 3. And after the zygote is there, sperms are collected 2 to 3 hours before egg collection, washed and kept in hams. ওই দরকার নেই তোমাদের। Then এক্ষুনি আসবো, this fertilized zygote ultimately form the blastocyst. কেমন? About 32 to 64 সেল হবে, 1 থেকে 2, 2 থেকে 4, 4 থেকে 8, 8 থেকে 16, তাকে আমরা বলবো blastocyst. Then this blastocyst is now transferred to the uterus via the vaginal route. আমরা ফিমেল ভ্যাজাইনা বা মাদার যে আছে তাকে আমরা ব্লাস্টোসিস্টটাকে ট্রান্সফার করে দিলাম, and uterus কে স্টিমুলেট করা হয় secretory phase by drugs or by auto, so that this is taken up by the uterus and baby is born normal. শুধু তফাৎ কি, fertilization occurs outside, sperm fertilization also occurs outside. তার জন্য আমরা বলছি in vitro fertilization বা assisted reproduction. এই যে এখনকার দিনে কিন্তু আগে অ্যান্ডোস্কোপি হতো, এখন পার ভ্যাজাইনা দিয়েও ওভাম কালেকশন করা হচ্ছে, দেখতে পাচ্ছ, ভেরি ইজি প্রসিডিউর। এটা হচ্ছে ভ্যাজাইনা, এটাকে বলে ফর্নিক্স। এখান দিয়ে একটা ছোট্ট দিয়ে আমরা ওভাম কালেকশন করছি। করে ওভামটা সাক ট্যাপ হয়ে এই কালচার মিডিয়াম আছে, এইখানে ওভাম এসে গেল। ভেরি ইন্টারেস্টিং। এই দেখো, এই যে টিউবটা, তারপর এখান দিয়ে এই যে এইভাবে আমরা দিয়ে ফর্নিক্স দিয়ে ওপা দিয়ে কালেক্ট করছি উইথ আলট্রাসোনিক গাইডেন্স। ফলস হচ্ছে না, আমরা তার সাথে আরেকজন আলট্রাসোনোলজিস্ট এখানে প্রোব দিয়ে দেখে নিচ্ছে প্রপার গেছে কিনা, প্রপার জাইগোটটাকে গ্রাফিয়ান ফলিকলকে কালেক্ট করছে কিনা, সুতরাং মিস হওয়ার চান্স খুব কম। এরপরে দেখো আর্টিফিশিয়াল পেট্রি ডিশে ওভামটাকে রাখা হচ্ছে, দেখেছো? এরপরে স্পার্ম দেওয়া হচ্ছে, এই যে দেখতে পাচ্ছ, এবং ফার্টিলাইজেশন হচ্ছে। এইভাবে ব্লাস্টোসিস্ট হচ্ছে। এইগুলি যে বলেছিলাম ড্রাগগুলি যে স্টিমুলেট করা হয়, এই সব ড্রাগগুলি দিয়ে স্টিমুলেট করা হয়। এফএসএইচ, গোনাডোট্রপিন রিলিজিং হরমোন, লিউটিনাইজিং হরমোন প্রোটোকল আছে, এগুলি এমডির জন্য, তোমাদের অত দরকার নেই, এমডি ক্লাস নেওয়ার জন্য তৈরি করেছিলাম। আচ্ছা, আরেকটা হচ্ছে, এটা একটু নামগুলি জেনে রাখতে শুধু, ইন্ট্রাসাইটোপ্লাজমিক স্পার্ম ইনজেকশন is a procedure in which a single sperm cell is injected directly into the cytoplasm of an egg. এখানে কি করছি আমরা, ওভাম রাখছি, স্পার্ম রাখছি, and they are allowed to fusion. Chances sometimes they may not fuse properly. আমরা ঝামেলায় যাচ্ছি, একটা স্পার্মকে হেডকে ডাইরেক্টলি বা পুরো স্পার্মটাকে ডাইরেক্টলি ঢুকিয়ে দিচ্ছি, হেডটাকে আমরা ডাইরেক্ট ঢুকিয়ে দিচ্ছি ওভামের মধ্যে, তাকে বলছে ইন্ট্রাসাইটোপ্লাজমিক স্পার্ম ইনজেকশন, ICSI। আরেকটা ইন্ট্রিস দেখো, with this method the acrosome reaction is skipped. acrosome reaction তো লাগছে না, আমরা ডাইরেক্ট ঢুকিয়ে দিচ্ছি। তো তার জন্য কি, only one sperm cell per oocyte, while IVF needs 50,000 to 1 lakh sperm লাগবে, এবং সেগুলি যদি প্রপার না হয় চান্স অফ ফেলিউর ইজ দেয়ার। সো ইট ইজ মোর ইম্পর্টেন্ট